Genome sizes vary across the tree of life. What you're looking at here on the x-axis is the C value of different genomes shown in picograms. So what we're looking at here is that a C value of 1 is about 1 billion base pairs. Or, I'm saying base pairs is BP. We can also call this 1 gigabase. And now on this scale of powers of 10, that means that 10 to the minus 3 would be 1 million base pairs. Or 1 megabase is another way of saying that same thing. Now, on the y-axis here, or ascending along the y-axis, you see all of these different organisms, and they're moving from top to bottom in order of complexity. So you have single-celled organisms down here, and then organisms that have many different types of cells and are more complex organisms. But as you can see, there's no real clear pattern in the size of the genomes as you go, especially when you start to think about the number of genes. So I'm going to point out some of the key players in biology. For example, E. coli is down here with the bacteria, has a genome size of about 4 million base pairs, and it has about 4,000 genes in that genome. Nematodes, C. elegans, they are about here with a genome size of 100 million bases. And with those 100 million bases, they have about 22,000 genes. I'm going to say 22K because I'm running out of room. And let's see over here with Drosophila. They're in the insects. Their genome size is also about 100 million base pairs. And with that, they have about 17,000 genes. My about sign looks a little funny there. I'll change that. And finally, there's us. Our genome size is about 3 billion bases. Seems to be about right there. And in those 3 billion bases, we have about 27,000 genes. So as you can see, the genome size is varying across all these organisms. It's not showing any clear pattern with complexity of the organism, especially when you think about the number of protein coding genes that are encoded in those genomes. So what is a gene? Let's diagram one and think about the parts. And here I'm specifically going to focus on a eukaryotic gene diagram, like a human gene. Here I've sloppily drawn some double-stranded DNA that is part of a genome. The first two elements of interest I'm going to say are the promoter and transcription start site. The promoter is where RNA polymerase is going to bind. I'm going to draw that here as a big yellow circle. And as we talked about last week, RNA polymerase is the enzyme that's going to transcribe DNA to RNA. So it will bind, this enzyme will bind at the promoter, and transcription will start to happen at the transcription start site. You can also have other regions that affect transcription, like enhancers. I've drawn an enhancer region up here, and what might happen is that a protein could bind that can somehow affect RNA polymerase, either recruiting it or helping transcription to happen. This enhancer might be really far away. So, for example, I'm going to say it could be thousands of bases away. It could be upstream or it could be downstream. And in some cases, enhancers are even inside of introns, which we'll come to in a second. This promoter and enhancer region are called cis regulatory elements. That is, they are DNA sequences or elements that regulate the expression of this gene in cis. Cis here meaning on the same molecule of DNA in the same region of the gene. What I've shown here in red is a protein binding that enhancer. That protein, in this case, is a transcription factor. Transcription factors are examples of trans regulatory. So I cannot talk and write at the same time. Transcription factors are examples of trans regulatory elements. A trans regulatory element also regulates the expression of a gene, but it does so in trans, meaning not from the same molecule of DNA. For example, here we have this red transcription factor thing that I've drawn but this is a protein. The gene that encoded this transcription factor is not in this gene. It's somewhere else. It might not even be on this chromosome. So this regulatory element, the transcription factor, is acting in trans, not on the molecule of DNA for this gene. In eukaryotes, once the RNA polymerase enzyme has been recruited to the DNA, it will transcribe RNA. I'm going to draw that here as a little single-stranded, RNA is single-stranded, molecule until it reaches a termination site. Then it will stop transcribing RNA. It will also reach a polyadenylation signal where it will just start adding A's, a big string of A's, to the end of that molecule. After RNA polymerase is finished transcribing the gene and has reached the termination sequence, we have a primary mRNA transcript. To talk about what happens next, I need to diagram a few bits of this primary mRNA transcript. First, I've drawn 3 exons. Exons are the bits of DNA in a transcript that will be expressed in the final gene product. If there are pieces of DNA that are being expressed, then here there are pieces that are not being expressed. Those are introns. Eukaryotic mRNA can have introns. These are intervening sequences that will not be included in the processed transcript. We call the action of getting rid of these introns splicing. So you can see here a spliced transcript that only has exons 1, 2, and 3. You can also get splice variants through alternative splicing. So you can see here I've drawn an example where exon 2 didn't make it into the processed transcript. The splicing happened to join exon 1 to exon 3, skipping over exon 2. And what this means is that one gene can encode multiple different products. This transcript is going to get processed in a number of other ways too. We already mentioned the poly A tail. It's also going to get a 5' methyl cap. These two parts of the transcript are important for downstream exportation of this transcript out of the nucleus. Again, this was just transcribed from DNA in a eukaryotic cell, therefore it was happening in the nucleus. This transcript has to make it into the cytoplasm and then be recognized by ribosomes for translation. Speaking of translation, we're missing a few important elements of this transcript that need to be labeled in order for us to understand what is going to be translated into protein. As we talked about last week, you need a start codon, here that's AUG, and a stop codon. I have randomly placed those within the exons of this transcript. Based on where I put that start codon and stop codon, you can see that we'll end up with a protein that spans from here to here. Of course, this protein has been translated and it's in an amino acid sequence now, but I'm placing it on this diagram to point out the fact that there is sequence up here. Let's see, what's a good color to point this out? And down here, regions that were not translated. And we call this the 5' untranslated region, or UTR, and down here the 3' untranslated region, or UTR. Just again to point that out, these UTRs are referring to the bits of sequence 5' of the start codon and the bits of sequence 3' of the stop codon. So bits of the gene that are not translated into protein. So let's start to think about everything that makes up the genome. We just learned about genes. Those are the most important, exciting bits of the genome, right? They must make up a lot of the human genome. Actually, when we look at it, we see that protein coding gene sequence only makes up about 2% of the human genome. If we add in intronic sequences, introns, the bits that get spliced out, and that's why they don't end up in the final protein, and all of those regulatory elements, the sequences where there were promoter sequences or sequences where RNA polymerase bind, or enhancers where transcription factors bind, we get to about another 30% of the sequence explained, but still not a lot of the genome is being explained by gene sequences. We can also consider the sequences that come from non-coding RNAs. These are RNAs that are not translated into protein at all. And these include things like tRNAs. tRNAs or transfer RNAs are the RNAs that translate RNA to protein by binding the amino acids to the appropriate codon. We have ribosomal RNAs. These are RNAs that make up parts of the ribosomes. We also have various RNAs like microRNAs and siRNAs, which are involved in gene silencing, and long non-coding RNAs like exist. That's that long non-coding transcript that's involved in X activation. This is not a complete list of all of the non-coding RNAs, but it does go to show that there are sequences in the genome that are functional, but aren't necessarily genes, protein-coding genes. The human genome and other eukaryotic genomes are also filled with a lot of pseudogenes. These are genes that were accidentally replicated in the genome. And over time, since they were a replicated copy of a gene and there was a functional copy available, they acquired deleterious mutations such that they're no longer functional. And they're kind of hanging out in the genome with unknown significance. The human genome also contains highly repetitive DNA sequences. Two of these, mini satellites and microsatellites, are just repeating sequences of different lengths. Mini satellites can be a little bit longer than microsatellites. But for example, a microsatellite might be a position in the genome where you just see ATAG, ATAG repeated over and over and over again. This is going to be important later when we talk about identifying genetic markers to compare populations and different individuals. Also, telomeres, or the ends of chromosomes, also have highly repetitive DNA sequences. These are typically 6 base pair repeats that just get repeated over and over and over again. Another large part of the human genome contains transposable elements. Transposable elements are dynamic sequences that can move around the genome and increase in number. And they do this by the use of transposase. So here I have diagrammed a simple transposon. So in blue is the DNA sequence surrounding the transposon. I'm going to always draw DNA as a single line. Just remember that this is double-stranded, right? And here I'm showing you a transposon in this DNA. The transposon is flanked on either side by sequence that is an inverted repeat, so they're in the opposite direction. And then within the transposon is an encoded transposase. What the transposase is an enzyme that's going to recognize those sequences, cut out the intervening sequence, and put it somewhere else in the genome. When the transposon jumps around in the genomes, it can do so by the cut and paste method, whereby the transposase recognizes the sequence, cuts it out of the genome, sometimes leaving little mistakes behind that I've indicated here, and then pastes it back into another position in the genome. This could be really far away or not too far away. One other mechanism is referred to as the copy and paste mechanism. Here, the transposase copies the transposon and inserts another copy somewhere else in the genome. Again, could be nearby, could be far away. But now you have double the amount of transposon that was there as before. So you can see how, especially this mechanism of transposition, is going to increase the copies of transposons that are in the genome. Also, I want everyone to be thinking, what are the potential consequences of this? We have a DNA sequence that's being randomly inserted throughout the genome, and we're acquiring huge copies of this sequence that doesn't help the organism. This isn't functional for the organism. Altogether, genomicists estimate that transposable elements make up about 45% of the human genome. And as a reminder, only 1 to 2% is estimated to be responsible for coding. So that leaves us with a lot of questionable material in the genome. And all of these numbers are big estimates. People are still working this out. But one surprising thing that's been showing up is that maybe as much as 80% of the genome is transcribed. That's really strange for a number of reasons. And there aren't any certain answers at this point, but I'm hoping that's something that we can talk about in class.